So we'll start our lecture today by talking about DNA replication. So DNA replication majorly is divided into three steps. Unwinding of the DNA, then adding short segments of RNA, then copying actually the DNA. For the unwinding of the DNA, it starts with a helicase, and this helicase breaks up DNA and breaks up, open the DNA through breaking up the hydrogen bonds that holds the two DNA strands together. That means it unzips the DNA. The DNA binding protein after that hold the single strand apart to prevent the formation of the hydrogen bond again, and to keep the two strands separated of the DNA by the help of the DNA binding proteins. The separation of the strands occurs originally at the start region of replication, which is called origins of replication. So that means you can find the multiple origin of replication throughout the DNA molecule. The second step is adding short segment of RNA. Actually, this is done by an enzyme which is called primase, and this enzyme adds RNA primers to the DNA. And these RNA primers actually, at that regions, the replication process starts. So why is RNA primers are added? Because actually the DNA replication cannot initiate de novo. That means the DNA polymerase, which will do the job of replication, cannot locate the region that will start to replicate from. So it needs a guide, and this guide is called the RNA primer. So once the DNA polymerase find the RNA primer, it will sit on the DNA strand and start to add the nucleotide and building up the copy of the DNA. So the third step is copying the DNA. DNA polymerase enzyme, as we said, binds to the RNA primers that is formed by the primase at the start of the replication. Then the DNA polymerase uses nucleotide to synthesize complementary strands to DNA. You must keep in mind that that always DNA polymerization occurs in one direction from 5' to 3' direction. And it's crucial for the process of replication. So replication occurs only in direction of 5' and 3'. And if you remember, there is a problem in the DNA because the DNA strands are anti-parallel. One is in the direction of 5' to 3', and the other in the direction of 3', 5'. So, for example, here you have the parental DNA. So you have here, let's have a pointer. So you have here the parental DNA, then melting occur. You can see the two strands are separated apart. Then the DNA polymerase start adding nucleotide complementary to the base to the nucleotide in the original strand, until forming two identical daughter molecules of DNA. So basically, the adding of the nucleotide is complementary to the original strand. If you look to the whole DNA molecules, so you can see the DNA start to melt at one region by the effect of the helicase. It opens up into two single strands, two parental strands. And these, as we said, the parental strands are held apart by the help of single strand proteins. And then the DNA polymerase starts to add the nucleotide complementary. You can see here, it's adding nucleotide, C to G, G to C, and it adds a complementary nucleotide until forming the double-stranded new DNA. Let's have a look how replication actually occurs. So as you can see, DNA is double-stranded, anti-parallel. Then helicase start its work by melting DNA. It opens up the DNA, forming what is called the replication fork, and it is the origin of replication. Then the short primers are added, represented here by the red segment. Primers are added, and then replication starts, represented by the green arrows. Let's talk first about the formation of the leading strand. As I told you, that replication occurs in the direction of 5 prime, 3 prime. So the lower strand actually, it starts with 3 prime. So a primer is added, then the DNA polymerase sits over the primer and then adds the nucleotide in this direction. So it's moving from 5 prime to 3 prime. And since the lower strand, let's see again, since the lower strand formation is continuous, that's why it's called leading strand, okay? So have a look, you can see as well the replication fork, and usually the replication fork, it's the region of 80 rich regions. So you can see the leading strand is continuous. That's why it's moving from 5 prime to 3 prime. The problem actually is in the other strand. Let's see why. So here you can see that at the lower strand, the primer is added. Then the DNA polymerase will sits here and start adding the nucleotide in this direction. And it's actually the correct direction because it's a 5 prime, then it moves in the direction of 3 prime, which suits DNA polymerase. But the problem is in the other strand, because once the other single strand is open, the RNA, the polymerization of the new strand cannot start from here, because if it's the primer is added here, the RNA primer is added here, the DNA polymerase cannot move in this direction, because actually it will move from 3 prime to 5 prime. So in this case, the primer is added near the replication fork, and then the DNA polymerase will move in the opposite direction because it's from 5 prime to 3 prime. And this lead to form short Okazaki fragments, and these Okazaki fragments is formed actually of RNA primer with a short segment of new formed DNA. Once these are formed, they are connected together by the DNA ligase. So another form of DNA polymerase will attach and remove these RNA primers, and then by the help of the ligase will join the segments of these DNA, forming a complete strand. And this is why, in that case, this strand is called lagging strand, because actually it's not continuous polymerization of DNA, but it is discontinuous formation of DNA. Let's see again. So helicase opened up, primer is added, DNA polymerase moves in this direction, then it opens up again, another RNA primer is added, moves in this direction. So as soon as the fork is open a little bit, a RNA primer is added at the replication fork. So let's see here. The primer is added at the replication fork, moves, then another opening. This will lead to another addition of RNA, another opening, more opening of the fork. Then we'll have another primer here, and the DNA polymerase will move in this direction. So now you can have 3 different Okazaki fragments. As I said, another form of a DNA polymerase will bind and remove these RNA primers, and then the DNA ligase will join these segments. So finally, you'll have a complete strand, but since it formed discontinuously, so it's called lagging strand. So again, this region is called replication fork, and and actually it is region of 80 rich regions. And you can see that the direction of fork is the movement, is actually the movement of the opening of the two double strands. Here you can see as well another figure representing the DNA replication. So you can see here that the helicase unwinds the parental DNA strands. Then you have a single-stranded binding protein that stabilizes the two strands to prevent them from joining back again. Then you have the leading strand, this one which is formed continuously in the direction from 5 prime, 3 prime, while the lagging strand is formed discontinuously, in which a primase adding a primer here. Then the DNA polymerase moves in the direction of 5 prime, 3 prime to form a short segment. Then a primer, here is primer, and forming a short segment. These Okazaki fragments are soon joined together by the help of DNA ligase. As we said, after the RNA primer is removed, this RNA primer is removed, the DNA ligase will join these segments, forming the complete growing of the lagging strand. And here is actually animation of what is actually, or how it's actually the DNA replication occur. Okay, so you can see here, you have one strand that is formed continuously, which is the below one, and the other strand you can see formation of segments of Okazaki fragments. That's why it's discontinuous. So let's talk about the RNA and protein sensors. So actually, from the figure, you can see that you have a DNA that it can undergo replication, as we have just said. DNA as well can do transcription to release, to copy genes from the DNA into a messenger RNA molecule. So the DNA code is translated into RNA code. Soon after that, this messenger RNA migrate from the nucleus to the cytoplasm to do translation, to do a protein, which actually give the trait, give the phenotype of the gene. So the transcription occur only in genes in which the RNA polymerase, in this case, bind to the promoter region, which is the non-coding region, which is up before the gene sequence. And this RNA polymerase will unwind the DNA double helix and will copy only one strand of DNA into RNA. As well, the RNA polymerase copies a DNA in the direction same as DNA polymerase, which is 5 prime, 3 prime. But the use in these cases, in this case, the U nucleotide instead of T. At the end of the gene, because every gene have a start point and the end and termination code, there are termination sequences that terminate RNA polymerase transcription. So the RNA polymerase and the newly formed RNA, they are released from the DNA, and the RNA strand actually, which is formed, which is called messenger RNA, it is now, it's still immature messenger RNA, so it must be called pre-messenger RNA or simply pre-mRNA. You need to understand that multiple copies of messenger RNA can be transcribed from each gene. So it's not only transcription, one single copy of messenger RNA. You can, genes are transcribed into multiple copies of messenger RNA. And soon after that, the messenger RNA, once translocated to the cytoplasm to be translated on the ribosome, each messenger RNA can be translated into multiple number of proteins, of the same proteins actually. So here again you can see this is the gene, this is the start site, and this is the termination sequence. So actually this is the region of the gene. The region before the gene is called the promoter sequence, in which the RNA polymerase sits on that promoter sequence and will move over the DNA to transcribe. So after that, there is unwinding of the DNA. You can see the RNA polymerase sits over and unwinds the DNA, and as well by the help of helicases. If we enlarge this region, you can see here messenger RNA starts to be formed, growing. If we enlarge here, you can see that the RNA polymerase, this whole circle, on top of the unwinded part of the DNA, and you can see it starts adding the nucleotides A and U to tRNA on the template strand. And the newly formed pre-messenger RNA will emerge. You can see as well that the end which is free, it is the end of 5'. That's meaning that the RNA polymerase is moving in the direction of formation of the new messenger RNA in the direction from the 5' to the 3'. Soon after that, once the RNA polymerase reaches the termination sequence, everything is released and you have a completed pre-messenger RNA, and you have the RNA polymerase again free because it might be used for the transcription of another gene. You need to understand that the process of transcription actually consists of initiation, elongation, and then termination, where the RNA polymerase reaches the termination sequence. What about messenger RNA processing, and why there must be messenger RNA processing? And actually, this is important in eukaryotes, not prokaryotes. So if you remember now, there are transcription of DNA into RNA, pre-messenger RNA, and actually the RNA processing, it is a process of converting the pre-messenger RNA into mature messenger RNA or mRNA, which have to be done before the migration of the messenger RNA into the cytoplasm for the process of translation. So the processing, because the initial messenger RNA produced, which is the primary transcript, the pre-messenger RNA, the immature messenger RNA, are not fully functional. So it needs a series of modification before these primary transcripts are ready for the process of protein synthesis. And actually, this modification of the messenger RNA processing actually it's a three different steps: the RNA splicing, the polyadenylation, and the addition of 5' cap. So this pre-messenger RNA must be modified by these three processes to have a mature RNA. So let's look closely to this DNA segment of DNA. You can see that this is the gene, and usually genes consist of regions called introns and exons. So introns, actually, it is the region that it is non-coding. Non-coding means that it will not give proteins when it's translated. So any genes is consisting of exons and the introns, a number of exons and introns depending on the gene. And the gene usually ends by a termination sequence, as we have just said. And upstream of the gene, you have a promoter sequence that help to identify the gene for the RNA polymerase when it sits over this promoter to start to transcribe the gene. You can see some enhancer elements of DNA sequences way above the genes or way upstream the genes, and this as well facilitate the process of transcription. So this DNA segment can be transcribed. You can see now you have the primary messenger RNA, pre-messenger RNA, and it have exons and introns. So now this primary or immature RNA have to be processed in order to be mature and functional to be translated in the cytoplasm. So you can see here that the three processes, RNA splicing and polyadenylation and addition of 5-cap already occur. So you can see by the end, you can see a number of adenine have been added. That's why every messenger RNA will have a polyadenine tail, and that's why it's called the polyadenylation. While up the front at the 5' end, so polyadenylation occur at the 3' end. While at the 5' end, you can see addition of guanine triphosphate. That this structure is called a cap, which is added on the 5' end of the messenger RNA. And this is a second modification. While the third important modification, it is the removal of the introns and the splicing back and splicing in the process of splicing, so that the messenger RNA now is only formed of exons, because as we said, these introns are not coding and they're no use for the process of translation. And in this animation, you can see that, so long. So you can see here in this animation, the process of removal of the introns and splicing back the exons. So actually, this is the third modification. So to summarize, any messenger RNA, after being transcribed from DNA, it have to be processed. So the processing actually, it is 3 steps: RNA splicing, polyadenylation, and addition of 5' cap. So the polyadenylation, actually, it is addition of a series of adenine molecule at the end of the messenger RNA at the 3' end, while the addition of a 5 cap represented by guanine triphosphate is added at the 5' end. While the RNA splicing, actually, it is the removal of the introns, which is the non-coding regions, and the stitching back the exons to form a functional coding segment of messenger RNA. So let's see the another important aspect of processing, which is alternative splicing. So scientists have found that every gene can produce a messenger in different forms of messenger RNA based on a very peculiar process, which is called alternative splicing. So in other words, the gene can produce different messenger RNA and protein product from the same genes. So you can see that from the figure, it is actually the same genes, exon 1, 2, 3, exon 1, 2, 3. It's the same genes. But this one gave this large messenger RNA, and this one give a small messenger RNA. So in this case, this will give a short protein, and this will give a longer protein. So we can understand now that some genes can give a different form of messenger RNA. As well, we can understand, if we look closely, exon 1 and exon 3, for example, these are exons, and this one, the exon 2, all here, when transcribed, they are treated as being, all of them are exons. But in this case, you can see that one of the exons, especially exon 3, have been treated as if it is an intron. So it was removed during the process of splicing. And this created another version of the messenger RNA that can be used for that can produce a different protein upon translation, and can be used for a different function. Generally, the major difference between prokaryotes and eukaryotes, the prokaryotes have transcription occur in the cytoplasm since there is no nucleus, while in eukaryotes the transcription occur in the nucleus. The transcription and translation in prokaryotes can occur together because there are no nuclear envelope. But here, the RNA have to be processed in the nucleus or modified in the nucleus before being migrated or exported to the cytoplasm. So in prokaryote, there is no processing is needed, but as we said, in eukaryote, processing is needed to increase as well the RNA stability. So let's see here another very good representation of alternative splicing. So you can see here, let's have a pointer. This actually the DNA sequence, and you can see it have 1, 2, 3, 4, 5 exons represented by colors. And then you have an RNA that is transcribed from the whole gene. You can see this actually, the arrow here represented the initiation of the gene, or the initial site of the gene. So it is transcribed. So you can see it have now a pre-messenger RNA. Why? How did I know that it's pre-messenger RNA? Because it still have exons, and these regions between the exons are actually the entrons. So now we have immature RNA. It needs to do alternate splicing, but here we will see alternative splicing. So in some types of cells, these 5 exons are exist. So removal of all intron. So you can see this messenger RNA will have all the 5 exons, and it will be translated to a protein A, whatever the name of that protein, in a special type of cell. While in another cell, you can see that exon 3 have been treated as if it was an intron, and only 4 exons are formed in the messenger RNA, and this one will be translated into a protein B, which is different from protein A, only from the region of the exon 3. And then you have in another cell type, exon 4 have been treated as an intron, and upon translation, it will give a third version of the protein, which is a protein C. So you can see that alternative splicing resulted into 3 different types of protein in 3 different cells. And this is the main advantage of alternative splicing. So let's see what is the importance of alternative splicing in this very good example. Okay, so as you already know now, that alternative splicing will generate different proteins from a single eukaryotic gene, from the same gene. As we know, in contrast to bacteria and archaea genes, the vast majority of genes in multicellular eukaryotes contain introns, multiple introns, non-coding regions. And the presence of these multiple introns in these eukaryotic genes permits the expression of multiple related protein, because they are so somewhat similar from a single genes, as we said, from the alternative splicing. So in higher eukaryotes, actually the alternative splicing is a very important mechanism for production, although they are different form of protein, but they are still isoforms. So it is similar to some extent in a sequence of nucleotides, only different in some parts, in different types of cells. For example, let's take an example, fibronectin protein. So fibronectin protein, actually, it is extracellular adhesive protein. So it's a protein, it's a kind of protein that help it is a sticky protein that it can stick to the places inside our body. So this extracellular adhesive protein found in mammals and secreted by 2 types of cells, fibroblasts and hepatocytes, which is the liver cells. And this protein is a very good example for alternative splicing. So actually, in fibroblast, because this fibronectin is coded in these 2 types of cells, in the fibroblast, the fibronectin messenger RNA contain 2 important exons, which is called E3A and E3B. And these 2 important exons in fibroblast will encode a protein domain or protein region or a protein part of that of the whole protein that enable the fibroblast to adhere to extracellular matrix. While in hepatocytes, the fibronectin lacks these A3A and E3B exons. So while the processing of the messenger RNA of the fibronectin gene in the hepatocytes, the A3A and E3B will be treated as if they are entrants and will be removed from the mature messenger RNA. So this will result in a fibronectin protein secreted by hepatocyte into blood that these fibronectin will not have the adhesive property that exists and cannot adhere to other cell type, and this will allow this protein, this fibronectin, to circulate in the blood. Let's see in the figure. So generally, fibronectin is more than 20 different, it's not only 2 isoforms, it's 20 different isoforms of fibronectin. You have 20 different versions of the fibronectin protein. And these, the 20 versions that is already have been identified up till now, each encoded by alternatively spliced messenger RNA composed of unique combination of fibronectin gene exons. So generally, you can see the fibronectin gene is consists of many exons. All these different colors are exons, either yellow, green, red, purple. All of them are exons, and the regions which appear like railroad between them is the entrance. You can see the entrance, and you can see where the A3B and E3A is located. In the fibroblast, you can see that the messenger RNA, this actually the messenger RNA which is formed, contain the 2 exons, which is E3A and E3B. While in the fibroblast, in the hepatocyte, the liver cell fibronectin messenger RNA lacks these 2. So the importance of these 2 exons, actually, when they are translated, when this messenger RNA is translated in the cytoplasm, these 2 exons will encode a specific sequence of amino acid that will help the fibroblast to adhere to the extracellular matrix. While for the hepatocyte, while it's lacking these 2 exons, so the fibronectin secreted, the fibronectin protein secreted by hepatocyte will circulate in the blood and it will never adhere. So let's talk about the basics of gene expression control. So the gene expression, actually, it is the production of messenger RNA by a cell. All cells of an organism contain the same gene. So how and why are skin cells different from brain cells or liver cells? Because actually cells can regulate or control the gene they express. They select which gene they express in each specific cell type. So for the gene regulation, how genes are turned off in response to different signals. While the transcription regulation, it controls the amount of messenger RNA transcribed from a particular gene. So RNA polymerase cannot bind to promoter region without the presence of transcription factors that help the RNA polymerase to locate the gene of interest that need to be transcribed. You can see as well enhancer sequence, which is sequences of DNA that combine proteins called activator, and these help as well in the process of transcription. You can see as well a repressor sequence that can bind to another set of protein to decrease the rate of transcription. All of these are modes of transcription regulation, either enhance the transcription or repress the transcription. So in this example, you can see here, let's have a pointer. So in this example, you can see here, hold on. This is the gene. Let's have a okay. This is the gene. This is the start region of the gene. And you can see this is a promoter sequence. And as I said in the previous slide, the promoter sequence is a sequence which is upstream of the gene, and it is used by the RNA polymerase and the transcription factor to identify the gene of interest need to be transcribed. And you can see in the promoter region, there are regions of a TAT box, so that's mean AT-rich region of a sequence of DNA. And you can see another region that is rich of a CAT box, CAAT box, and this actually specific sequence that is all located in the promoter region upstream of the gene. And you can see as well the enhancers can be located far away of the gene and have a specific sequence. And these enhancers, once activator or hormone or any protein that is bind to this enhancer can, once this activator bind to this enhancer, it will enhance the transcription of that gene. So you can see now that once the activator binds to the gene, this will bring the promoter nearer to the RNA polymerase, and the RNA polymerase will bind to the promoter region to start transcription. You can see from the figure, the enhancer with the activator, it will help in the formation of the RNA polymerase complex with transcription factor over the promoter region to start the process of transcription, and the transcription initiation complex is formed, and so the RNA polymerase start to work. So bacteria actually use a structure called operons to regulate gene expression. So operons actually are clusters of related genes that is located under the control of a single promoter. So that's different from the eukaryotes, because as we have just seen, in eukaryotes, every gene have one promoter, single promoter upstream of the gene for the binding of the RNA polymerase. While in eukaryotes or bacteria, they have a number of genes, a number of related genes, all of them have one single promoter, let's say. So what is the operator? Actually, it is the region within the promoter, it is inside the promoter, and it regulates gene expression in response to nutrients requirements. And we will see an example for that. So let's see an example, the Lac Operon. So what's Lac Operon? Actually, Lac Operon, it consists of three genes which encode to three enzymes, and they are very important in the process of transport and the breakdown of lactose. So here you can see, for example, the three genes are, you can see Z, Y, and A genes here. This is as three genes. You can see the promoter region, and here is the operator region. And you can see here the RNA polymerase sits above the promoter region and then moves over the DNA to transcribe these structural genes to form three different enzymes upon translation. So here you can understand that it's the major difference between eukaryotes, because here you have three different genes. All of them are under the control of one promoter, while in eukaryotes, each gene have its own promoter. So lactose actually, the molecule is the operator activator. So what's the operator do? As we said, it regulates the expression of this operon. So the whole structure here, which is consists of the promoter, operator, and the three structure gene, it's called Lac Operon, all these structure, and Lac for lactose, of course. So if no lactose is found, there will be a Lac repressor protein, Lac repressor, which will bind to the operator, Lac operator, and will blocks the movement of RNA polymerase to form the messenger RNA, and hence there is no need for the transcription of these genes again. So this operon only works in case there is lactose in the medium of the bacteria, and it needs to metabolize this lactose to get energy from this carbohydrate molecule. So in case there is no lactose, if there is no lactose, it will be a waste to transcribe these genes, because these genes actually generate the enzymes necessary for the breakdown of lactose, the transport and the breakdown of lactose. So if no lactose is found, there is a protein which is called the Lac repressor, will bind to the Lac operator and will prevent the RNA polymerase from doing its job, which is transcription. And actually, the Lac Operon, as we said, consists of three structural genes. The Lac Z, which will form the galactase, the Lac Y, which will form the enzyme primase upon translation of the messenger RNA, and the Lac A, which will form the enzyme acetylase. And all these three enzymes, as we said, are important for transport and the breakdown of the lactose. So let's see again here how this happened. So actually you have the I region here, and this actually why I, because it's inhibitory. So this one actually generating messenger RNA at that region, which will be translated into protein. This protein, once take the 3D conformation shape, it will form the repressor protein. So actually this repressor protein, if there is no lactose in the medium of the bacteria, this repressor protein is supposed to bind to the operator. Once this repressor protein binds to the operator, there will be no transcription of these enzymes, of these genes to form the three different enzymes. But what actually happens when there are lactose in the medium? The lactose in the gross medium or the medium around the bacteria will bind to that repressor protein. The binding of lactose molecule to this repressor protein will prevent the binding of this repressor protein to the operator region. This will enable the RNA polymerase to transcribe the gene. So messenger RNA will be formed, which will be translated by the ribosome in the cytoplasm to form the three different enzymes that help in the transport and the breakdown of the lactose in the medium of the bacteria.